Okay, a very good morning to all of you. And before I start, I need to put a little disclaimer that my talk is going to be a little different from whatever you have been hearing in the past 3 or 4 days. My talk, the work which I'm going to present today is going to tilt heavily towards the applied part of IUTAM. So where I'm going to use the principle of mechanics to understand some biological processes. I'm pretty confident that all of us have gone through this process in our life, where we have developed from a single cellular embryo towards a multicellular engine. So basically, that is the process of development, which is primarily growth and the deformation of biological tissues. And as you can see that because this involves growth and deformation, that means mechanical forces are definitely going to play some role. And this was presented in a very brilliant form around a century ago by Darcy Wentworth Thompson in the book On Growth and Form, where he described the physical principles behind the biological form of different microorganisms and the scaling principles. But in the past 2 or 3 decades, the research of mechanical forces in the biological development has taken speed because of the new technology which have come in the recent past. So what I am going to present in this talk is a special tissue known as epithelial tissue. That is the tissue which covers almost all of our organs, including our skin and including the skin as well as the internal organs. And I will present its structure and specifically the components which are essential if we want to model the mechanical behavior of the epithelial tissue. Then I will describe the Vertex model representation for the epithelial tissue and its physical characteristics, and whatever we understand from this physical modeling, I will use that to study a specific biological problem. So first of all, as I mentioned that the development is nothing but the deformation of the tissue. So here I am just showing 2 examples where you can see that during the development of a fruit fly, there are huge deformations and flows of cells taking place. And this is another example where there is because of the contraction of the adjacent tissue, the wing of the fly takes its shape. And the tissue of the wing is also an epithelial tissue. And so first of all, I will describe the structure of the epithelial tissue, which is the center of the work. So epithelial tissues are the tissue where cells are arranged in a specific manner, and depending on the arrangement of the cell and the shape of the cell, they are divided in different types. So if the cells are flatter, they are called squamous tissue. If they are tall, they are called columnar tissue. And what we are interested in is the columnar tissue, where the individual cell have a cylindrical shape and they are perfectly packed. So if we look closely at the epithelial tissue, what we observe is that constitutively we can safely assume that it is homogeneous, but there are some structures which are present at specific locations in the epithelial tissue which provide it polarity, which is known as apicobasal polarity. So if we focus on the apical side of the epithelial tissue, we find there are 2 main structures which are going to be most important for the modeling of the epithelial tissue. So here I'm just showing the top portion of the epithelial of this tissue, these three cells which are neighbor to each other. So black outline shows the membrane of the epithelial tissue, and then there is another structure which I have shown in red, that is known as the actomyosin cortex. That is, there is another structure which is just beneath the cell membrane, and these are actin and myosin are the protein polymers which can actively generate stress to shrink the size of the apical side. So this is an intracellular machinery. That is, each cell has its own actomyosin system. In addition to that, there is another system which is not intracellular, but it builds up because of the intercellular interaction between the neighboring cells. So here I'm showing this by the help of these green bars. So there are some proteins known as cadherin proteins which are present on the surface of the cell. So when two cadherin molecules from the neighboring cells come close to each other, they form a very strong bond that leads to adhesive interaction between the neighboring cells. So even though there is some apicobasal polarity in the epithelial tissue, but in almost all of the cases, except a few which have been discovered recently, the topology of the apical and basal side is found to be same. That is, whatever structure of the epithelial cross section on the apical side is, it is same on the basal side. There are a few exceptions. I should say that. Therefore, if we assume that the topology of the epithelial tissue is same on the apical and basal side, that gives us a very big advantage that we can model it not in 3 dimension, but we can easily model it in a 2-dimensional system, and that's what we are going to do. So yeah, and there is another point is that this adhesive proteins which are present in the cell, they are present only on the apical side. They are not present on the whole of the lateral side. If they are present on the complete lateral side, that may give some that will give some that is going to give different behavior. And I hope that by the end of this talk you will realize what is the effect of its presence at the apical side. So if we look at the cross section of the epithelium, I'm sure many of you present here can easily recognize that this resembles very highly with the 2-dimensional dry foam, this dry foam has a very defined mechanical behavior that for small amount of shear strains it behaves as a solid, but if sufficient amount of stress is applied, it starts to flow. And in the modeling of the dry foams, the equilibrium for the static foams, the equilibrium configuration is obtained by minimization of the length of the contact lines, because the line tension is the force which is the most dominant there. So taking motivation from the dry foam, we can construct some energy functional for the epithelial tissue, which incorporates all the forces which are present in the form of actomyosin contraction or the intercellular adhesion. So we are going to model the epithelium as a confluent tissue. That means the cells are completely packed. There is no hole or gap, and we assume that the system is quasi-static, because for most of the biological processes, the deformation of these tissue is very slow. I will give you the numbers in the later slide. So this assumption is this assumption is quite safe. Then we model the epithelium as a network of vertices and edges. So I'm showing here three cells, which are three cells, and the nodes are labeled as I and J, and the cells are labeled by the Greek alphabet alpha, Greek letter alpha. And we can also, and same as the foam, we can obtain the equilibrium configuration for the epithelial tissue also by minimizing the energy functional, which is given by here. I will explain term by term. So the first term, as you can clearly see, that corresponds to the area elasticity, and the second term corresponds to the contraction which is imparted by the actin myosin, and here L is the perimeter of any given cell. And third term corresponds to the cadherin adhesion, which is there between any two neighboring cells. And here Lij is the length of the edge connected by the vertices I and vertex J. As you can see that, as I have mentioned, that actomyosin network which is present in the cell, that tries to constrict or contract that tries to minimize the apical area of the cell. That means this constant gamma needs to be positive, so that the size of the cell is, so the perimeter of the cell is minimal. On the other hand, the interaction, the cadherin interaction, that is the adhesive interaction between the two neighboring cells, tries to expand the cells, and that means that this parameter lambda here takes a negative value, and this is quite opposite from the dry foams, for which there is no actomyosin contraction present. There is no such machinery, and there this adhesive term takes the form of a line tension that takes a positive value. So with this framework, what we can get is, if we minimize this energy functional for any given initial configuration of cells, simple minimization of the energy functional can only give us a change in the cell shape and change in cell size, but that is not going to give us change in topology of the network. For that, we also introduce in the model the T1 transition, that is the exchange of neighbors. So here, which is explained here. So here, if you see in the first figure, the two cells marked by red dots are neighbor, but by some process, which can be some external force or some internal machinery of the cell, they go far apart, and the yellow cells become neighbors of each other. So that is known as T1 transition. So in the model, what we do when we simulate the model, if any edge goes below a certain length which we pre-specify, then we switch the neighbors. If that change of neighbor is favorable, it will remain. If it is unfavorable, it will go back. In addition to this T1 transition, which is also present in foams, there is also another T2 transition which is specific to the epithelial tissues, that is cell death and cell division. So in the tissues, depending on the biological context, cell also undergo death or delamination. So that means a cell which is present in the tissue can just disappear. So once we have these ingredients, we can calculate the force by assuming the quasi-static nature. We can calculate the force on every vertex by the functional derivative of the energy, and the velocity of each vertex. Then we can advect the vertex of the epithelial network by considering an overdamped dynamics by the force which we have calculated. So for simulation, what we do is we first give some seed points, some quasi-random seed points, and use the Voronoi tessellation to obtain the initial configuration, which may not be the equilibrium configuration. So here in this initial tessellation, the forces on each vertex will be non-zero. So then we equilibrate and obtain the equilibrium configuration with the appropriate values of the contractility and the adhesion parameters. So if we consider that the contractility and the adhesion are same in all the cells, then when we equilibrate the tissue, we obtain different configuration, and here I am showing two extreme cases. So when the actomyosin contractility is the dominating parameter over the adhesive interactions, what we observe is that the tissue takes a hexagonal packing, and on the other hand, when the adhesive interactions between the neighboring cells are dominating, that means the adhesion is very high as compared to the actomyosin activity, then the cell packing becomes irregular. Not only that, we also see that in this case where we have hexagonal packing, the cells have smaller cross-sectional area, and in case of irregular packing, the cross-sectional area is a little larger, and that is not unexpected. That you can easily see from the energy functional. Now this is the equilibrium configuration, and we can also so these are just two extreme cases. So we may also have some intermediate cases where the cell size and the cell packing will be somewhere in between. So what happens if we apply some shear on the epithelial tissue? So here I have shown you two extreme cases. So I will show you the response of the tissue when the shear is applied in these two systems. So this is the plot of shear stress versus the applied shear strain for these two cases, and so the blue curve which is shown here, that is for the cells which have hexagonal packing, and the green curve is the one where the cells have irregular packing. And as you can see that the response of the hexagonal packing of the cell is highly stiff as compared to the one for the irregular packing of the cell. But we also observe that when the shear strain is applied, the stress in the hexagonal packed cell increases, then it also shows a sudden decrease. So if we look closely at the geometry of the cell during the application of the strain, what we see is that there is a sudden T1 transition which takes place. So here I have shown four configuration of the tissue at the 4 time points. As you can see here, if you look closely, then the two black cells are not neighboring to each other when we are starting the experiments. But as the shear is increased, then the T1 transition takes place, and now in the final configuration, the cell packing again becomes hexagonal and the two black cells are neighbor to each other. Then again the same cycle takes place. However, on the other hand, in this hexagonal packing of the cell, the T1 transition takes place at once for the almost whole of the tissue, but in case of the irregularly packed tissue, the T1 transitions are taking place throughout the process of the shearing. That's why we do not see any shear. That's why the shear stress is completely vanishing. You may also see that there is some negative values for the shear stress that is coming because of the numerical implementation, because we have set some critical value of the edge length for the T1 transition. So if we reduce the edge length, critical edge length for the T1 transition, then this negative values go away. So what is the message here from this simple experiment is that when the actomyosin contractility is high, then cell has smaller size, it has a very regular hexagonal packing, and it behaves more like solid. That means it has a finite shear modulus. On the other hand, when the cell adhesion is dominating over the contractility, then cells have irregular packing, they have larger cross-sectional area, and they also behave more like fluid. And I need to emphasize here the relation between the cross-sectional area and the solid and fluid-like behavior, just because the experimental result which I am going to show, the experimental system there, the measurement of the rheological properties, the direct measurement of the rheological properties is very, very difficult. So what we are going to do, we are going to take the cross-sectional, the measurement of the cell area as a proxy for its rheological properties. So in the next, what I am going to present you are the experimental results where we will use the knowledge we have at this stage from these modeling studies. So before I go to the experimental result, I should first tell you what is the system. So what we are interested in is the development of a fruit fly. So it goes through 3 different stages, goes from larva to becomes pupa during its development, then finally metamorphosed to an adult fly. And during this transition from pupa to adult fly, it forms all of its appendages and organs, and the organ which we are interested in is known as thorax, which is also shown here, which is a beautifully symmetric organ. But and it is perfectly symmetric organ despite the fact that it does not develop as a whole, as a single entity, but it forms by the merging of two contralateral halves. They come from the 2 sides and then meet each other, and despite this, it finally gives us a very symmetrical organ. So here I'm just showing those 2 contralateral halves of the future adult thorax. So these are colored by some proteins which are present on the cell boundaries. So as you, first of all, what you can see is that the shape of these 2 halves are not identical. That is, when they come close to each other, here also it is not identical, but at the end, in the end product, what we get is a perfectly symmetric organ. Yeah. So as you can see, so this is the picture of the thorax during the early developmental stage, and this is at some later developmental stage where the 2 halves have moved closer to each other by the activity of these cells which are marked by red. So the cells which are marked by red, they contract and pull the 2 halves together. And the boundary between and in the biology literature, the boundary between the 2 tissues are known as leading edge, which is labeled in the future slides. So just to give you some numbers, what is the speed and the size of the system? So here I'm showing the whole process. How does it take place? So this time is with reference to some biological time, that is some biological event, and with respect to that, this event where the 2 halves are there. So one half is this, and other half is this, and at the end of 6 hours, that is around 1.5 hours, the whole process takes place. So from this stage, in 1.5 hours, the whole merger takes place. And So typical cell size in this tissue is roughly 10 micrometers square, and from the simple calculation we can get that the average speed of cell movement during this process is very small. So if you compare this movement with the, let's say, bacterial motility, it is roughly 100 times slower. That's why when I said that we can assume the epithelial tissue to be quasi-static, this is the motivation that the is very slow. So at each point we can assume that the tissue is in equilibrium. So to look closely at the rheological behavior of this tissue over time, as I should recall you that in the modeling experiments which I showed you, there was some relation between the size of the cell, the cell size and the rheological property of the tissue. So when we looked at the size of the cell, we see that the cell sizes are not uniform in the two halves of the migrating tissue. Instead, what we see is during the very early stage of the process, the cells are larger here, then again smaller here, then again some large cells are there, then again small. But at the end of the process, most of the cells are very large. So that means what we can say from here that the mechanical properties of the tissue is not homogeneous, but at the end of the process, the cells are larger, tissue is more fluid-like. So then the question comes, what is regulating this process? What is regulating this change in the cell size, and in turn change in the mechanical property of the tissue in a location-specific manner? So in order to see, so there are two parameters which I described in the model. One is the actomyosin contractility, and other is the adhesion between the cells. And there are two separate proteins which are responsible for these two mechanisms. So here I'm just showing the size of the cell as a plot. So red cell are the larger one, and the blue are the smaller one. And here is the expression level, the level of the proteins which is responsible for the adhesion of the cells. And as you can see that this adhesive protein is roughly uniform over the tissue. And these are the proteins which are sort of canonical in the biological system. They are the simple the cadherin proteins which are present in almost all of the tissue. So this is the first candidate which is looked at. So if the expression levels of this protein is uniform, but still we are getting some non-uniform sizes of the cell over the tissue, that means this may not be the candidate. And the second is the actomyosin, which I am not showing here today, but that also shows us a similar pattern. So that means we need to look at some other proteins which may give us similar behavior. That means they may either alter the adhesive properties of the cells, or they may alter the contractility of the cells. So I will not go into detail how did we come to these particular proteins. That's a completely biological question. But there are two proteins which are present there in these specific tissues. They are known as fat and daxious. And what is interesting about them is that they form intercellular heterodimers. That is, if the fat is present on one cell and daxious is present on other cell, then they are going to meet and form a heterodimer like the cadherins. Fat from one cell and fat from other cell is not going to form an adhesive interaction. So that's why it's called heterodimer. They do not form homodimers. And so here is just a representation of these heterodimers, which leads to an increase in the adhesive property of the cells. So here fat is represented with the red bar and daxious with the blue one. and as you and if the fat of this cell form a heterodimer with the dextrose of this cell, and vice versa. So if we want to study the effect of fat and dextrose, we need to modify the vertex model formulation such that it takes into account fat and dextrose also. So one way would be to just increase or decrease depending on the activity of these two additional adhesive protein. Just modify the value of this adhesion parameter and just get with it. But what is seen here in these systems is that fat and dextrose are not uniform. That is, we cannot assign a single value to this parameter. That is, it may have a different value depending on the location in the tissue. So we have to take the adhesive heterodimer into account. So the way we have done it is the final value of adhesion is the basal adhesion, that is the basal level of the adhesion which is present in all the cell, and on top of that there is also the adhesion which comes due to the fat and dextrose heterodimers. And for that we also need to know the level of heterodimers which are formed between the cells, and that is governed by this particular equation, which is not very difficult to understand. So this is just the rate of heterodimer formation is nothing but the freely available fat and dextrose, and there is some cooperativity involved. Next is whether similar to the adhesive protein expression which I have shown you earlier here, which is roughly uniform throughout the tissue. We also need to look at how does the fat and dextrose, that these two adhesive proteins are expressed in the tissue. As you can see here, the top two panels show the expression level of fat and dextrose at some intermediate stage of this process. So here you can see that the fat is high in this region and low in this region, and dextrose is high in this region and low in this region and the topmost region. And when at the end of the process, also we have a gradient of fat and dextrose over the tissue, but the expression level of fat and dextrose overall is also gets increased. So we also need to incorporate this detail into the vertex model. That is, how we do it is we specify the level of fat and dextrose. So here the blue shows the level of dextrose in a particular cell, red shows the level of fat in a cell, and this is not the initial configuration. In the initial configuration we start with a perfectly rectangular tissue with all of the cells having same size and same rheological behavior. But when we incorporate the fat and dextrose both in the vertex model, what we observe is that we observe the similar cell size distribution as we have observed in the experiments. That is, at the intermediate stages we see different cell sizes depending on the location in the tissue, but at the end of the closure we see that the sizes have increased throughout the tissue. This shows that during the intermediate stages of the process, not only the cell sizes but the mechanical properties of the tissues are also not uniform. However, at the end of the process, the cells are uniformly large and more fluid-like. So, what is the consequence? I mean, why is this happening? What is the use of this in this biological system? So, due to lack of time, I will not not explain this slide, and instead I will just go to this process where in the experiments one of the fat and dextrose, that is one of the protein, has been removed from certain regions of the tissue. So the top row shows the control case where nothing is disturbed. In the middle row shows where the dextrose has been removed completely. As you can see here, the expression level of fat are maintained, and the bottom row shows where the fat has been completely removed from the tissue. And here you can see that here we observe different cell sizes depending on the tissue location, but here the cell sizes are more or less uniform. And this actually reflects in the final outcome, that is the final animal. And that you can see here that when there is no disturbance, the organ is perfectly symmetric, everything is fine. But when any of this is disturbed, when any of these adhesive protein is removed, then we see that there is some gap on the thorax in the middle. Not only that, the symmetry of the organ is also lost. If you look at closely, then the hair patterns are also quite symmetric here, but they are not as symmetric here. And similarly, in the dextrose also there is a big gap there, that is known as spaced thorax in biology. So again, if I go back to this aspect, why is this change from the cell size distribution, which is not uniform, to a large cell sizes at the end? So this is not really an experiment, but a speculation which we get from here, that if you remember the starting experimental results where the two halves were completely asymmetric, but at the end the organ has to be symmetric. That means when the two halves are merged, which are not yet symmetric, they need to be fluid enough so that they can deform and form the final symmetric organ. So that is the purpose of we speculate that is the purpose of this fluidization of the tissue. So from here I will just summarize. So we have seen that epithelial tissues can demonstrate wide range of rheological behaviors, and the behavior of foam is just behavior of two-dimensional dry foam is just a special case of them. And in the real biological system, this epithelial rheology is regulated by regulating the expression levels of proteins which are responsible for the adhesion or the contractility. Now, the one thing which we are really working on and hope to share in the future is the influence of activity on the rheology. So I have not shown you some part here where the tissue which lies in between the two migrating halves, that actually has some activity, that is it contracts in an oscillatory manner. So what we are trying to see is how does that contraction changes if we put some contraction in the epithelial tissue, how does that changes the rheological properties of the system. So finally, I need to thank few people. So some of the experiments were done by Thamarai in the lab of Professor Pradeep Sinha. Thamarai is there if anyone wants to get the details about the experiments. And I also need to thank people from Grenoble, especially Shauqi and Abdus Samad, who were really helpful with some of the discussions. And finally, thanks to all of you for the patience.
From the part before you introduce the two proteins, the fat and dextrose, okay. More related to the shear when you apply the strain.
Do you want me to go there?
Okay. It's fine. I can ask. So I just want to understand: does this material show both solid and fluid-like behavior at different strains, or does it show only solid-like or only fluid-like, depending on the stage of development?
Okay, depending on, so it depends on the expression level of the proteins. If the contractile proteins are higher, it is going to behave like a solid for low strains. If the strain are high enough, then then this system is also undergoes the plastic flows. Yeah, as I shown in using the T1 transitions.
Yeah, because you showed two extreme cases, right?
Yes.
One where one is higher than the other,
Yes.
and one is, and each of them showed different behavior with
Yes.
respect to strain.
Yeah.
But is it possible to have like a single model where it goes from solid-like to fluid-like behavior?
Actually, this is a single model. We are just changing the values of the parameters.
Oh, okay, but without changing, you cannot go from. Sorry. Okay. Thanks. Yeah.
Joe?
I had a bit of problem understanding the Vertex model. Could you put up the equation for the energy? The question, while you're finding, yes, the question is, I assume that first term, A naught, is the unstressed, the area you would have due to growth.
Yes.
A minus A naught.
Yeah, A naught is just some preferred area.
It's the Eshelby stress equivalent of inclusion. But it raises the question, I don't know what material is being added here. Are the faces membranes or the edges? What's growing? The edges?
Okay, so here, so this model of the tissue is just by the vertex and the connecting nodes. So there is no, we do not consider any material inside of it. We just consider the epithelium to be a network.
The edges contain all the material.
Yes, you can say that.
So I would have put L minus L naught instead of the area. That's, in other words, you're stressing
No,
edges, incrementally.
yes, but there is one thing that if you recall that this is just a two-dimensional representation, but in reality the tissue is three-dimensional. So this area conservation is not from this material, but this area conservation comes from the volume conservation of the three-dimensional tissue. So this area can expand by reducing the height of the tissue.
Maybe we can discuss it further. I really don't understand.
So it may involve bending, you mean? It includes bending and stretching of the membrane, or just stretching?
No. In this formulation, there is the bending is not there, but stretching is there.
Okay, and you're just saying that when you go from three-dimensional projected to two, that stretching turns into an area change?
Exactly. Yeah.
Okay, so some geometric argument.
It's a
Yeah.
geometric representation.
Additional.
Yeah. We have time for maybe one more question, if there is. Okay, go ahead.
So when you apply shear, you show some dislocation.
Some?
I mean, stress is increasing and drop.
Yes.
Then kind of layer is shifted.
Yes.
And
Yes.
I noticed many layer is shifted at the same time. Is it true? Or only one layer is shifted?
Okay. So that's a very interesting question. It actually depends on how you are in the simulation, how you are going to apply the strain. So one way would be to keep one of the boundary free and just move the upper boundary. Okay. So if you do that, then most of these transitions take place near the moving boundary, and that is also known in the foam community. But in the numerics, if you apply, let's say, a fine shear strain, then the result which I have shown, where there I have applied a fine shear, and that leads to a shift of all the rows. Yeah.
Okay, let's stop there and thank our speaker.